Hello students, we have already studied about the male reproductive structure. We have also studied about the structure of pollen grains, and we have also discussed about the formation of the male gametes. So now it is time for us to start the second essential wall of the flower, which is the what? Yes, which is called the gynoitium. In front of us, we have the unit of the gynoitium. Do you know what the unit of the gynoitium is called? The unit of the gynoitium is called the pistil. The pistil is also known as the carpel. Now, the number of carpels in the flower can vary. It can be 1, it can be 2, it can be 3, or it can be many. Depending on the number of carpels, the flowers can be categorized as monocarpellary and polycarpellary, also called as multicarpellary. Whenever we are using the term monocarpellary, you all know mono means what? The word mono signifies one. So whenever I am telling that the flower is monocarpellary, I mean that in that flower there is the presence of only one carpel. Now, when the number of carpels are more than 1, that means it can be 2, 3, 4 or any number, but it should be more than 1. So, we use the term polycarpellary or multicarpellary. Now, the term poly or multi means what? Many. So, whenever there is the presence of more than one carpel, we will be telling it as a polycarpellary or a multicarpellary condition. Now, the multicarpellary condition can further be of two types: syncarpous condition or apocarpous condition. In front of us, we have the structure of the carpel of papaver. Please observe that the carpels are attached to one another, or you can say that the carpels are fused with one another. And whenever the carpels are fused with one another, we use the term syncarpous condition. Syn, the word syn indicates fusion. On the other hand, we have the carpel of Michelia. Please observe that in this structure, the carpels are not attached to one another, or we can say the carpels are free. And whenever the carpels are free, we call it as the apocarpous condition. Now, let us study in details about the unit of the gynoitium, which is called the pistil. So, in front of us, we have the structure of the pistil. If you observe the structure of the pistil, you will see that there is the presence of three major parts. This part on top is called the stigma. Below the stigma is the style, and below the style is the ovary. The stigma acts as the landing platform for the pollen grains, meaning that when the anther matures and they dehisce or break open, the pollen grains are released, and these pollen grains will first come and land on the stigma of the pistil. Now, below the stigma is the presence of a slender structure which is called the style. The style is actually the connecting link between the stigma and the ovary. And below the style is the presence of a bulged swollen structure which is called the ovary. Now, if you take a transverse section of the ovary, within the ovary, what do you observe? You observe that there is the presence of an empty hollow structure which is called the ovarian cavity. And within this ovarian cavity is the presence of a multicellular parenchymatous nutritive tissue which is called the placenta. Can you observe the placenta? It is very clear for us. Within the ovarian cavity, there is the presence of a structure which is known as the placenta. Now, pay attention, my dear students. We are discussing a very important point here. Arising from the placenta is a structure which is called the Megasporangium, and which you commonly call it as the ovule. Let's have a look. So within the ovarian cavity is the presence of the placenta, and from the placenta arises some structures. These structures are called the ovule, also called the megasporangium. Now the number of ovules can vary. It can be 1, 2, 3, 4, or it can be many. For example, it is 1 in case of rice and wheat, and it is many in case of the papaya and watermelon. Now, my dear students, please remember that this question is very commonly asked. What structure or which structure arises from the placenta? From the placenta arises the ovule or the megasporangium. Is it clear to all of you? Now, the ovule is attached to the placenta with the help of a thin structure which is called the stalk, and this stalk of the ovule, with the help of which it is attached to the placenta, is called the funicle. So within the ovarian cavity we have the presence of the placenta, the ovule or the megasporangium, and the presence of the stalk which is called the funicle. Now, let us talk about the structure of the ovule in details. So, in front of us, we have the structure of the ovule. The first structure that we observe here is the stalk of the ovule. Can you name the stalk of the ovule? Yes, we have already discussed it. The stalk of the ovule is also called the funicle. The body of the ovule is called the nucellus. What is the body of the ovule called? The body of the ovule is called the nucellus. Now, please observe that the entire body of the ovule is surrounded by protective layer, and these protective layers are called the integuments. The body of the ovule is surrounded by protective layer, and these protective layers are called the integuments. The integuments will encircle the entire body of the ovule except at the tip of the ovule. Can you see that at the tip of the ovule the integuments are absent, and the place where the integument is absent is known as the micropyle. Just opposite to the micropyle, can you observe it clearly? Can you understand it? Just opposite to the micropyle is the presence of the chalaza. The chalaza is said to be the basal part of the ovule. Now, another very important point which you need to remember, that the funicle is attached to the body of the ovule at a certain region which is known as the hilum. So, at the hilum, the funicle is attached to the body of the ovule. Now, you must remember that from the nucellus arises the female gametophyte which is called the embryosac. Now, depending on many factors, we can categorize the ovules into various categories. First, we will be categorizing the ovules based on the number of integuments. I know this is not there in your NCERT, but you have to know this because you often get neat questions from this section. So, depending on the number of integuments present in the ovule, you can categorize the ovules as unitegmic ovule, bitegmic ovule, and ategmic ovule. Unitegmic. Can you tell me what does the word uni means? Yes, the word uni means one. So, how will you define an unitegmic ovule? We say an ovule to be a unitegmic ovule when there is the presence of only one integument. It is often found in the gamopetalous families. Next is the bitegmic ovule. When the ovule will be having two integuments, we will call it as the bitegmic ovule, and it is generally found in the members of the polypetalous family. And ategmic ovule, ategmic means that in these ovules integuments will be absent, and this is a very special condition which is generally found in case of plants like Loranthus and Santalum. Now, let us again categorize ovules based on the position of the micropyle, the chalaza, and the funicle. The first type of ovule under this section is the Orthotroposovial. if you see the structure of the Orthotropous ovule, you will observe that in the Orthotropous ovule, the micropyle, the chalaza and the funicle will be lying in one straight line. Can you see it clearly? Observe this very clearly, that in case of an Orthotropous ovule, the micropyle, the chalaza and the funicle is lying in one straight line. And this is generally found in case of Polygonum. Next is the Anatropous type of ovule. In the Anatropous type of ovule, you will see that the ovule is bent at 180 degree. Meaning that in an Orthotropous ovule, what did you observe? You observed that the micropyle, the chalaza and the funicle was lying in a straight line, but in Anatropous ovule, you will see that the ovule is bent at an angle of 180 degree. So because of this, what do you see? The micropyle now lies very close to the funicle. Remember, the Anatropous ovule is the most common type of ovule. It is found in case of 82% of the angiosperms. Next, we have the Hemianatropous ovule. The Hemianatropous ovule is the ovule in which the ovule will be bent at an angle of 90 degree to the funicle. Observe clearly, the ovule is bent at an angle of 90 degree to the funicle, and this is commonly observed in case of ranunculus. After the Hemianatropous ovule, we have the next type of ovule which is called the Campylo Tropisovule. The Campylo Tropisovule is also bent at an angle of 90 degree to the funicle, but the only difference is that in this case the micropyle is bent slightly downwards. In Hemianatropous ovule, the micropyle was just normal, but observe the diagram of the Campylo Tropisovule. Here the ovule is bent at an angle of 90 degree to the funicle. What is the difference? The difference is that the micropyle is bent slightly downwards. And this is generally found in case of Leguminaceae and Cruciferae families. After this, we have the Amphitropous ovule. Observe the diagram of the Amphitropous ovule. In the Amphitropous ovule, the ovule as well as the embryosac is curved as a horseshoe. The shape is resembling that of a horseshoe, and this type of ovule is generally found in case of Lemna and Poppy. And now let us talk about the last type of ovule under this category, which is the Sarcinotropus ovule. The Sarcinotropus ovule is bent at an angle of 360 degree, and because of such a bend, we see that the funicle is encircling the total ovule. This type of ovule is generally found in case of Cactaceae family like Opuntia. Now, let us start with the process of Megasporogenesis. Megasporogenesis means that it is a process of formation of haploid megaspores from diploid megaspore mother cell. So, the process in which the diploid megaspore mother cell undergoes meiosis or reductional division to form haploid megaspores is known as Megasporogenesis. Now, let us talk about this topic in a bit more details. So, if you observe the ovule, one cell of the nucellus towards the micropylar end of the ovule will become large, and it will have a dense cytoplasm and a prominent nucleus. Observe clearly that one cell of the nucellus towards the micropylar side is becoming large. It is having a dense cytoplasm and a prominent nucleus, and this cell is said to be the megaspore mother cell. This megaspore mother cell will undergo meiosis or reductional division. So, megaspore mother cell is what? It is diploid, and this diploid megaspore mother cell will undergo meiosis, and it will be producing 4 haploid megaspores. Out of these 4 haploid megaspores, 3 of the megaspores towards the micropylar side will degenerate. and only one megaspore towards the chalazal side will remain functional. And this functional megaspore will later on form the embryosac, which is the female gametophyte. So the functional megaspore is involved in the formation of the embryosac, which is the female gametophyte. So the process in which there is the formation of the embryosac from a single megaspore is known as monosporic embryosac formation. Monosporic is indicating the fact that this embryosac has come out or has been evolved from a single megaspore. So this type of megaspore formation is known as a monosporic embryosac formation. Now, let us talk about the embryosac formation in details. Here we will be studying about the monosporic embryosac formation in details. So, in monosporic embryosac formation, as already discussed, 3 of the megaspores degenerates from the micropylus side and only one megaspore is functional towards the chalazal side. And this functional megaspore will now undergo nuclear mitotic division and it will be forming the 2 nucleated stage. Now these 2 nuclei that are formed, they will migrate towards the opposite sides, or we say as the opposite poles of the cell. And these 2 nuclei will undergo second round of mitosis and they will be forming the 4 nucleated condition. This 4 nucleated stage will undergo third round of nuclear mitosis and they will form the 8 nucleated stage. So my dear students, if you observe carefully, you will understand that during this mitosis, every time there is the karyokinesis or the division of the nucleus. But the division of the nucleus is not instantly followed by the formation of the cell wall. So, the cell wall formation will only take place after the 8 nucleated stages established. And once the cell wall is formed, then the typical embryosac will be developed. Now, let us understand the distribution of the cells in a typical embryosac. You see that out of the 8 nuclei that are formed, 6 of the nuclei are surrounded by the cell wall. But 2 nuclei are present in the center and it is the nuclei of the largest cell of the embryosac, which is the central cell. Now moving towards the micropylus side of the embryosac, you see that at the micropylus side there is the presence of 3 cells which together comprises the egg apparatus. The egg apparatus consists of the 2 synergids and the egg cell. If you look at the structure of the synergids, then you will see that the synergids contain special thickenings, and these thickenings are known as the filiform apparatus. And the filiform apparatus plays a very important role. The role of the filiform apparatus is to guide the entry of the pollen tube inside the embryosac. The filiform apparatus actually will be synthesizing certain chemicals with the help of which the pollen tube gets the guidance as to where it should enter the embryosac from. And then along with the synergids, there is the presence of the egg cell, and the egg cell, as you all know, is the female gamete. Moving towards the chalazal side. In the chalazal side, there is the presence of 3 cells which are known as the antipodal cells. So, a typical embryosac is 7-celled and 8 nucleated condition, meaning there is the presence of 3 cells in the egg apparatus, there is the presence of 3 cells in the chalazal side which are the antipodals, and there is the presence of 1 large cell which is called the central cell. So together how many cells are present? There is the presence of 7 cells. But remember, as the largest cell of the embryosac, which is the central cell, contains 2 nuclei, so the total number of nuclei present within the embryosac is 8. So that is why we describe a typical embryo sac as a 7-celled and 8-nucleated condition. Also remember that it is often asked in your question paper, what is the arrangement of nuclei within the embryo sac? Remember that 3 nuclei are present towards the micropylus side, that is the 2 nuclei of the synergid and 1 of the egg cell, and then the central cell has 2 nuclei and the antipodal cells present towards the chalazal side again have 3 nuclei. So if you are asked to write the arrangement of the nuclei within an embryo sac, you have to write this as 3 plus 2 plus 3. You cannot change the arrangement. The arrangement of nuclei within an embryo sac is 3 plus 2 plus 3. You cannot write it as 3 plus 3 plus 2. You cannot write it as 2 plus 3 plus 3 because this arrangement is very specific and it is 3 plus 2 plus 3. I hope the structure of the embryo sac is very clear to all of you.